Yeah. Okay. Good morning, everyone. Wanted to welcome you all to the Movement of Life quarterly meeting for fall 2023. I'm Matt Ogburn. I'm a senior scientist here at the Smithsonian Environmental Research Center, leading our Fisheries Conservation Lab, and I'll talk a bit about that research later. But we're excited to be able to have this hybrid meeting. We have some folks here in person at CERC that will be sticking around after the meeting for lunch and a tour and happy hour at the end of the day. But also very excited to have a number of people joining us online to be able to share their work from afar. So I'm just here to welcome everyone, to say hello, and I'm going to turn things over to Lacey for a few comments about the Movement of Life program more generally, and then we'll move into our research talks.
Perfect. Thank you, Matt. Lacey, are you looking at your like what screen are you seeing on the Zoom? Oh, I see. Yeah, I see that. You see that? Okay. That's correct, right? Yes. Okay. And just a couple housekeeping things for people in the room. We've got lunch planned at 12 o'clock in the garden in front of admin. The weather's good today. So those of you that placed orders, it will be there when you get there. And if you didn't, please bring your own or just come join for the conversation. There will be cookies. And it's providing cookies. There's a campus tour from 1:30 to 3:30. We still have space for anyone that would like to join. I think we're planning on Autumn, Lynn, Catherine, Jimena. Is Ronaldo? I don't. You're on. Okay, Ronaldo. And okay, great. Yeah, Candace. And I think that's everyone. But if you weren't named and want to join, talk to Kim or Matt to make sure you get to the right place. We're going to meet at the circle near the front entrance of Matthias Lab out there. Yeah. And then 4 o'clock, a happy hour at the Reed Pavilion here on campus. So there's a campus map attached to the last email about the meeting, but also ask us where to go. Okay. And those of you online, thank you for joining us. We, Kim and I will be monitoring the chat and questions. So if you have questions, just raise your hand or type them in the chat and we will we'll get to you. So, and we want this, all Movement of Life meetings are meant to be, am I looking at them with that? Mm-hmm. Okay, great. Are meant to be conversational. So we welcome a lot of participation, Q&A, and we're going to try to save time for that, especially at this meeting, is to really promote collaboration with attendees. So, and then just a few announcements before we begin. There's a interesting Wild Labs group poll about biologgers. I don't know what the full intent of that is, but that biologging group is really active and a great place for information about all sorts of tracking tech questions. And I figured this group would have a lot to contribute to that. So there's a link to this in the agenda. Please check it out. Chris Fleming's also hosting a full day CTMM workshop at the Wildlife Society annual meeting, which is November 5th. So those are always, they fill up fast, but it sounds like there may still be spaces. So check that out. He also wanted to share that their MOVE design paper recently came out in Methods in Ecology and Evolution. It's a shiny app used to evaluate sampling design for animal movement studies. So it's, I think, really badly needed. And I haven't gotten to play around with it, but I know if it came from Chris, it's going to be good. So check that out too. There's a link to that paper in the agenda as well. And a request for content for the transmitter. If you haven't sent me anything and would like it to be included, the transmitter is our monthly newsletter. It was on a break the last few months while I was out on leave, but we're back and I'm going to get another one out in the next week or two. So send me, major conservation achievements, any announcements you want to make The only key is that it needs to have a link, like a web link, because we don't have a lot of room for text in the newsletter. So if you have a paper, it needs to have some kind of press release that we can direct people to as well. Okay. Then I'll stand over here for my show. I guess.
That should move your slides.
Okay. Okay, and since we have guests here from really around the world, namely Peru, I wanted to give a general overview of movement of life again. Also, we just haven't given this to our group in a while, so just a really brief intro about what we do and why we do it. So I am the program manager and also an ecologist with Movement of Life, and do I point it somewhere?
At the computer in the corner, it's gonna work. The big stacks.
Oh, that.
Kim, you might just need to click on the slide too. With the mouse. Yeah, bring the mouse over onto the. But now it should work.
Okay, good.
So if you want to do your own slides, just click it on. Yeah.
I think the clicker will work now too.
Okay.
Maybe not. Okay.
Oh, yes,
There we
okay. Okay, so we are a pan-institutional initiative. So that means that we include research groups from all across Smithsonian, primarily at the Conservation Biology Institute here at CERC, and also at STRI, and Migratory Bird Center is one of our large partner that's also at SCBI or NCCBI. And I know I don't need to convince this group why movement is important, but sometimes it's helpful to sort of zoom out to the big picture again. And really, it's just that movement underpins all kinds of important ecological processes. It connects ecosystems. It drives key biological processes and underpins healthy communities. However, we still don't know a lot about how global change is affecting the movement of species on Earth. So, but the Smithsonian, a lot of dramatic pauses here. The Smithsonian has actually been in the game of animal tracking for maybe longer than many people realize. So over 100 years, starting really with Paul Barch at the National Zoo. He did the first bird banding in North America. Also, Smithsonian was involved with the culling of Monique the space elk. I think Movement of Life is very familiar with this story at this point, but if you're not, Google her. She's the first GPS collared animal or mammal, and a really interesting story. And then Tuck Hines, of course, here, our resident star.
He looks exactly the same.
He does. He led the first telemetry study of molting in arthropods here at CERC. So there's our witnessing history. We've since grown into a real global network of collaborators and research sites. Our team is led by a collaborative of scientists from across the Smithsonian. And we really have three key pillars to our approach. So one is doing basic and applied research. So using animal tracking data to inform conservation, working really closely with local communities to make sure our data is being put to use, and training the next generation of movement scientists. Just a quick overview of some of our key questions that we try to answer. We're studying animals across land, air, and sea. And a really unique feature of our program is our link with the National Zoo and captive collections, and the opportunity to both trial alternative attachment techniques and also be involved with species, the reintroduction of species to the wild. As I mentioned, a key part of our work is really working with local stakeholders to put our data to work. So some very important outcomes, just a small selection, or reducing elephant, or discovering the issue of elephant poaching in in Asia, and reducing ship strikes on whales in South America. This is Hector Guzman's work. Also, stewarding big data for conservation. So we have some really impressive kind of convening mechanisms and groups that are collating data from partners all around the country and the world. Right here at CERC2, Atlantic Cooperative Telemetry Network, led by Matt and Kim, and the Shorebird Science and Conservation Collective with Candace and Autumn. And then we have an extensive array of training, fellowship programs, internship programs. So if this is new to you, reach out to me if you're interested in these opportunities. And of course, Smithsonian is the world's largest educational and research complex. So we have some really unique opportunities to share the power of animal tracking data with the public, both in exhibits and digital media. So some really fun. Check out the birdhouse at the zoo. And that's all. I'll take questions after everybody. So I think with that, we're just gonna dive into our speakers, yeah? And Jimena is gonna do our introductions.
Okay. I don't need to stand up, right? No.
You can,
I can do it
they
from
can
here?
see you, I think.
Okay. Okay, I'll go ahead. Okay, so our first speaker is Dr. Alex Hearn. Alex, welcome. He's a professor of biology at the University of San Francisco de Quito in Ecuador, and he's the founding member of the Miramar Network. Alex is a marine fisheries ecologist with 20 years of experience working in Galapagos. He's a founding member and current board president of the Regional Research Network Miramar. And he recently led the multi-institutional team of biologists to develop a blueprint for improved conservation of the open waters outside the Galapagos Marine Reserve, a process which resulted in the creation of the new Hermandad Reserve in 2022. So welcome, Alex. So nice to see you.
Thank you, Jimena, and it's so nice to be part of this. It's nice to put some faces to names as well, so that's great. Yeah, so I wasn't really sure what to present here. So what I thought I'd do is just give a brief overview of Miramar as a network and the kind of tracking work that we're doing that led to the creation of Hermandad or expansion of COCOS and kind of how it fits into a regional context. So yeah, forgive me if it's pretty basic, but I'm more than happy to go into details if there's a question session afterwards. So let me see if I can bring up and share my screen. So you should be able to see my screen. Is that right?
Yes. Yes.
Yeah, awesome. Okay, great. So and now I can't. There you go. So Miramar is a network that operates basically in the heart of it is the Eastern Tropical Pacific. In fact, one of the founding members is Hector Guzman. I saw he was mentioned on one of the previous slides with the whale work in around Coiba in Panama. So it really began about, gosh, just under 20 years ago, when several individual scientists working in this region, I was based in Galapagos at the time, working on fisheries, and most fisheries in Galapagos back then were benthic. So, you know, we had the sea cucumber wars back then, we had spiny lobster and bacalao. And there was a strong push by local fishermen inside the marine reserve to allow long lining. And we were just trying to find some information about, well, how's that going to affect the shark populations in the marine reserve? And we realized there was absolutely nothing known about sharks in Galapagos other than there are plenty of sharks in Galapagos. So that was really my entry into doing some research on migratory species. And also it kind of switched that vision that we had Galapagos became a marine reserve in 1998. So for the first 10 years, most of the work was looking from Galapagos inwards. So my work was based on these, coastal benthic resources. Some of the species are endemic. We were concerned about invasive species, but it was always Galapagos inwards, which I think was a direct translation from the terrestrial work. On these isolated islands, and really thinking about how longlining might affect sharks in the reserve forced us to just switch our point of view and start looking at the role of Galapagos regionally for these highly mobile species. And I've shown here several species. My own work is more on sharks and molars, but within the region we have people working on tuna, we have people working on blue whales, so all sorts of species. But the idea is that these are highly mobile. Some of them may have defined migratory routes, and so you need to look beyond your own study site in order to be able to really come to grips with their conservation. And around the same time, this political vision was created between Costa Rica, Panama, Colombia, and Ecuador called the CEMAR, which is the Marine Corridor of the Eastern Tropical Pacific. And it was largely at the time driven by Conservation International with their seascape idea. But it became a political initiative led by the governments of the 4 countries. And the idea of the CEMAR is kind of this lofty vision of sustainable use of ocean resources across the region and protection of endangered species. And it's morphed over time. I mean, you'll see this map for a start. I often question this map in the middle because there's this gap here of international waters that they seem to have taken control of. And I'm always told that, well, this is a conceptual map. It's not a physical map. But with this idea of the CEMAR, having scientists based at Coiba, Cocos, and Malpelo, and Galapagos that you'll see here, it made sense to start talking and sharing information. So really this Migramar network was born out of that need to feed this political process. And so, I mean, this is just some quick photos of the work that I do in Galapagos, but a lot of this, it's the same kind of techniques used elsewhere. So we're studying, nursery sites for endangered species such as hammerheads. We did a lot of acoustic telemetry back in the early days, and the data that we collect at the moment is being hosted by OTN, which I'm sure many of you are familiar with and work with. And then, of course, we moved into satellite tracking where possible to start looking at migratory routes. And then added to that, you start asking questions about, well, what is driving these movements? And right at the end here, you see a photo. This is we've after many conversations over many beers and whiskeys, decided that it might be possible to do underwater ultrasounds on free swimming whale sharks at 20 meters depth and in 3 knots of current. And when the hangover wore off, we realized that actually it might work. And so we've been doing ultrasounds of this parade of large female whale sharks that come through Galapagos each year. This is an interesting one because it's always nice to have on our team people with different views. And I think at this point, we're suffering from the Mulder and Scully complex, which I call it, which is when some of us really want to believe that they're pregnant, and half of us, after having done 22 ultrasounds on free swimming whale sharks and there's no evidence of it, are starting to look at other hypotheses. But it's really interesting to see how these contrasting visions on the same project can really help push the boundaries of what we're doing. And we've added some citizen science in there too, because what we're finding is that so a lot of the tracking work is great to understand core habitat use, whether there's particular routes from which they're moving. But a lot of the work that we do is inside marine reserves, and a lot of these marine reserves don't really have long-term monitoring programs. And so we're trying to develop cost-effective monitoring of some of the megafauna, and a lot of this is done through the scuba dive community as well. So after, we all started working in our different sites and realized that, if we shared technology, if we all used Vemco receivers and the same tags, and if we shared data sets, then we can get a lot more information out of it. And in fact, the first animals we ever tagged back in 2006 in Galapagos, we tagged 18 hammerheads. 2 of them turned up at Cocos Island pretty much the same day, 6 months later. So that was our first evidence of connectivity between Galapagos and Cocos. We're now looking at 20 species over 11 countries, and really we've moved from becoming an informal network of people that just call each other and send emails with data sets to a formally established organization. That was after about 10 years of working informally. So in 2016, we became established as a 501c3 organization, able to receive funding and with a core team. Everyone else, we all belong to our own institutions. I'm not an employee of Migramar. I work at the university here in Quito, but Migramar does have a small core team of employees, and that gives us great flexibility because everyone's working in their own organization spread across the Pacific. We're able to act simultaneously across the region, and we're able to adapt to those fluctuations that come with different funding cycles without disappearing. The main thrusts are research, obviously, research to understand the movements and spatial ecology of threatened species in the region. We translate that into policy advice. We work very closely with national governments as well as this regional CEMAR vision. And then I can't emphasize enough capacity building. I mean, we are in part of the global south in this region. We and we very much want to ensure that where we don't have the technical capacity, if we ally ourselves with partners who can bring them in, but that they can leave that capacity here when they go, so that we can then generate our own science. So a lot of training, both within our scientific community, but also with students. And again, as a professor at the university, that's one of my core jobs too. So here's just some brief, just some quick images. There's Hector there, who's a member of STRI. A lot of expeditions out at sea, a lot of collaborator expeditions. And hang on a minute. And so some of the benefits of Migramar or of an organization or network such as this is, data sharing, allowing us to use compatible technology, even sometimes getting discounts if we're buying in bulk, which is always good, ensuring that we're standardizing our protocols, and therefore we can visible over the last few years. Some of the challenges are coordination. It can be a little bit like herding cats sometimes when we've got 24 members spread over 11 countries. A lot of it has to do with trust as well when we're sharing information, project ideas. And so membership is always an issue about who we can bring in, who's going to have access to certain sensitive information. There's definitely a need for structure. This is something that I think we're now translating to other areas. The fact that you have everyone working in their own site, thinking locally, but you have a core team who 24/7 are thinking regionally, really allows us to push forward in leaps and bounds. And I think we're trying to show the political process that they need that too. And sometimes there can be conflict of interest, and you have to keep aware about mission drift too. But other than that, we're finding the experience to be very positive, and I'll show you why. So basically, with the information that we're gathering, information on migratory routes, information on the oceanography of the region, community structure as well. We're using BRUVs at the moment. And then for some reason, I can't change the slide. Hang on. Oops. There we go. So this is kind of some of the information you can see here down between Darwin and Wolf are two islands that are separated by about 30 miles within Galapagos. And we can start understanding how they work as a dual refuging system where you have hammerheads gathering there during the daytime. They leave at dusk, they come back at dawn. Presumably they're heading offshore to feed. They're there from June through to March, and then there's this absence, and that's when we're detecting them in Cocos. So we're able to really understand their behavior in and around these islands. And then on a larger context, this larger map just shows some of the movements that we've picked up with satellite tracking across the region from Revillagigedos all the way down to northern Peru. And so this information is used then to develop policy briefs. This is one that's very close to my heart, which is this idea of a swimway between Galapagos and Cocos, where we found now that several species are moving loosely associated with the Cocos Ridge between Galapagos and the Cocos Island Marine Reserve. And so we proposed the creation of this swimway, which would be a transboundary protected area in the ocean. And this has now been declared a Mission Blue Hope Spot. And my own work over the last couple of years has focused on the Ecuadorian side of this, where we now have some of that area is now take 60,000 square kilometers. And so we're working on these different swimways across the region and also developing one in Mexico. I just wanted to just finish up then. So back in 2010, the total area of marine reserves in the region was about 156,000 square kilometers. And as we've moved over the last dozen years, you can see those numbers ticking up. And we're now, this is actually already out of date. We're now at 720,000 square kilometers. And I'm proud to say that every single square kilometer that has been protected or declared in this region has been done so to some extent using the science generated by folks in Miramar. And that's where we see our impact, when we can see this before and after map and know that the science that we've done has contributed to this. So with that, I think I've probably exhausted my 10 minutes. So yeah, we'll leave you for any questions.
Thank you so much, Alex. It's really impressive. And I didn't say at the beginning of the meeting, but a key goal of this meeting is to just learn more about Miramar. There's obviously a lot of similar goals between our organizations, overlapping interests and complementary geographies. So we'd like to focus a lot of the conversation around how we might be able to work together more. And we have time for probably one question, I think, for each speaker right now, but we'll have hopefully some time for conversation towards the end.
Fantastic.
I have a question.
I was gonna say, I will have a question, but I want to wait until after a couple of the other speakers.
Okay, I think it's probably better for timing anyways. So, okay.
Yeah.
Yes. I can do next.
Okay, thank you, Alex. Next speaker, it's Matt Ogburn. I'm just gonna do the introductions for people online. So Matt leads the Fisheries Conservation Lab here at CERC. He specializes in the ecology, management, and conservation of marine and estuarine fisheries. He applies field and laboratory experiments, animal tracking technologies, models, long-term observational studies, and historical perspectives to address fisheries use issues, including sustainability of fish populations, altered food webs and trophic interactions, linkages between habitat and fisheries, invasive species, and climate change. Welcome, Matt.
Thanks, Jimena. I'm excited to be able to share a few things about some of the work that we're doing here at CERC Fisheries Conservation Lab. And if you want to follow us on X, formerly known as Twitter, we're at CERC Fisheries. And I'll comment a little bit sort of related to Alex's talk about our regional network, the Atlantic Cooperative Telemetry Network also. So yes, we're doing research on a variety of different types of things. A lot of it, but not all of it, is focused on animal movement types of studies. And our fisheries work is sort of in similar ways to what Alex was just describing for Miramar, is a lot about understanding the ecology of species that we fish for and the ecosystems they depend on, and then thinking about how to apply that information to fisheries management, conservation, and restoration. And through sort of those research activities, and some personal connections, we ended up with an opportunity a number of years ago to help sort of mature and evolve the local telemetry network on the East Coast, which is the Atlantic Cooperative Telemetry Network, which started back in 2005 or so, in sort of similar ways to what Alex described, a group of sturgeon researchers who wanted to start using acoustic telemetry to track their animals. and realized they were migrating out of their own individual acoustic telemetry arrays to other places and needed some way to start sharing that information among each other. And so for the first almost 15 years, that looked like more or less a Google spreadsheet that listed every tag. And Cam, I forget how many you said there were.
Like 19,000.
Yeah, so like grew to about 19,000 tags. So a giant Excel spreadsheet of tag IDs. So every time you went to one of your receivers and downloaded data, this map gives you a sense of the scale of how many receivers were on the East Coast of North America. You had to go look at that list, figure out who owned those tags, and write them all emails with their data. And if you were a researcher getting data, it came back in a different format from every single one of those people. You had to turn it into something useful. So with some funding support through the US Animal Telemetry Network, which started up officially around 2017, but there was probably a decade-long planning process to get to that point. We were able to sort of both take over leadership of that as some of the original leaders were ready to hand it off, and also working with the Ocean Tracking Network in Canada to build a functional sort of back end of our Atlantic Cooperative Telemetry Network or ACT network data portal, so that we could do automated data sharing in the same way that Nigrodmar does, working with OTN. And actually, if one of my tagged animals managed to swim all the way down there, automatic data sharing would happen to let me know that. So the ACT network now used to be pretty much the whole Atlantic coast of North America, but in the sort of current form, the ACT network covers approximately North Carolina to Maine. There's a group to the south called the PAC network that extends to the Caribbean, and then the Ocean Tracking Network in Canada. And we're able to share data automatically across this whole region. The map on the right shows just the ACT network receivers that have been deployed over the last couple of years. I'll show numbers in a minute, but each different color on the map is a different project. So this is just to give you a sense that there's a lot of different projects that together make a larger network. So currently there's 126 projects in our database from 180 researchers at 85 different institutions. So it's a very big network of people that we work with, and Kim is the data manager, so she's the one interacting with all these people. There's currently about 802 active stations and about 3,500 active tags swimming around there that are in the database, and there's more that aren't in the database. We're still sort of onboarding people, and yeah, there's new research going on all the time. But what's in our database spans all the way back to about 2002 or 2003 and covers 59 total species. And we have a new project that we just gotten funded for to help with one of the big new drivers of acoustic telemetry research on the Atlantic coast, the mid-Atlantic coast in our region, which is the soon to be very rapid development of offshore wind energy. There's a lot of studies going on with it, and not a huge amount of coordination between the different companies and their contractors and our network. Some of the contractors are members of our network and are facilitating that for the companies they're working with, but a lot aren't. So we're getting some funding to increase capacity, and I've just interviewed a couple people and need to make final decisions soon, hiring a postdoc to help with this project. But the ultimate goal of this, which is funded by NOAA's Northeast Fisheries Science Center, is to develop a national strategy plan that you know, for new people coming into acoustic telemetry, especially related to offshore wind, lays out how you get into it, what all the standards are, how data agreements work, and sort of how to play together. So we're excited about this new project. And I put some contact information here, but mostly put this slide up to show that there's a lot of different parties involved in the ACT network, from funders to collaborators to RPS, which provides the data science side of this and infrastructure. I want to highlight just a couple of research projects as well. One that we have going on right now that we're really excited about is tracking the migrations of river herring. We've been studying them in Chesapeake Bay since 2013. This one picture here is an alewife. And we just in the last couple of months got data back for the first full annual migration cycle for this species from anywhere on the Atlantic coast. So Kim, I think I'm going to need you to click this video and play it. So this is an animation put together by colleagues at University of New Hampshire, showing 50 tagged alewife. They'll disappear and kind of blip around a little bit. But as a group, you'll see at least one of them went all the way up to Canada, almost to the Bay of Fundy. And then in the fall, they start migrating back down and come back the following spring to spawn right back where we tagged them in the Choptank River, where Autumn Lynn is from. So, that's the first full annual migration cycle for the species, and we're very rapidly working on writing up a quick publication on this because a bunch of other people tagged them 1 year after us. Trying to get that out there. And then I just wanted to highlight, we built on some of that initial funding for that work in the Choptank River and also in the Rappahannock River, came from the Life on a Sustainable Planet program. And this is a picture of a couple of people, well, Rod Aguilar from our lab, Jack Ryan, and who's the environmental director with the Rappahannock tribe in Virginia, and Mark Fortune, who's the assistant chief of the Rappahannock tribe, tracking or tagging alewife in the Rappahannock River. And as a group, also with George Mason, we just got funding from NOAA to expand this work to 3 rivers in the Chesapeake over the next 3 years. That proposal was led by Henry Leggett, who's the postdoc in the lab. And the last one that I want to touch on briefly is sort of a synthesis work that's the collaborative project with the University of New Hampshire, Nathan Fury, who's presented at one of our quarterly meetings, and Mariah Livernoy, who's a postdoc with him, integrating data from a bunch of different things that we've tagged here in the Chesapeake. And so the species we've tagged have ranged from things like horseshoe crabs to several species of sharks and rays, striped bass, the alewife that I mentioned. So the inset map here shows the tagging locations. So they're either in Chesapeake Bay or the mid-Atlantic. The larger map shows the farthest detection of all of those things. So Canada to the Florida Keys. And then we're starting to, through this project, use standard metrics to describe those migrations. So in this case, it's maximum distance traveled, simple metric by species. So you can see some are going a long way, some are going very far. This is time on the x-axis and latitude on the y-axis. So you can see some of them are doing these big coast-wide latitudinal migrations and other ones aren't. And then we're looking at exploring how those different species are moving through different management jurisdictions so that we can help provide information on movements to fishery managers. So some like horseshoe crabs and common carp and blue catfish are pretty much sticking around where we tag them, and other ones are migrating all up and down the coast through different jurisdictions. And then lastly, we're trying to do a lot of education as well, and looking for opportunities for that. We had a former grad student, Sarah Mallett, who's on the call today as well, who worked with Smithsonian Science Education Center this summer, creating a curriculum for grade 3 students on protecting whales and their migrations, just as one example of that work. So I think we'll stop there. And but thanks for the opportunity to share some of our work here at CERC.
I think we're going to continue the trend of holding questions till the end. So go our next speaker.
Okay. Okay, our next speaker is Martin Benavides. Okay, Martin, yes, hi. Martin, he's a postdoctoral researcher at in the Biological Monitoring and Assessment Program. Martin is a marine biologist with a decade of experience studying shark and ray populations worldwide, particularly in the Americas. His research has used a combination of approaches, including spatial ecology, time series analysis, and molecular ecology to understand residency and habitat use, population dynamics, and population structure for a variety of sharks and ray species. Welcome, Martin.
Thank you. I just wanted to confirm that you can see and hear me okay?
Yes.
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 Okay. And are my slides showing yet?
Yes.
Yes.
Oh, they are.
Yes.
Okay.
Can you see
Great.
them?
But all I can see is a message saying your screen share is loading. But anyway, as long as you guys can see them, that's what I mean. I see them on another screen. Well, thank you for that introduction, Jimena, and thanks so much for having me. I wanted to talk today about a project that I was kind of in the right place at the right time to help get off the ground. I want to acknowledge the fact that a lot of the the conception of this study came from Jimena and Matt Ogburn. But anyway, we work as part of the biological monitoring and assessment program at this facility that you're seeing in the background of this slide, which it's a large artificial marine infrastructure. And as you can hopefully make out looking at the coastline, it's in an area that is largely devoid of any hard structure. So obviously that creates some really interesting ecological dynamics, which we are trying to understand, and this study forms part of that. But to give you some context, hopefully you're seeing a map now that shows a much wider area in Peru. Just to orient you, the slide I showed before, the picture is taken right there along the coastline. And just to point out that the B map, it actually encompasses a much wider range of habitats. You know, we work in the marine habitat, but as you can see, this strategic alliance includes also a gas pipeline that comes down from the mountains, and then at our facility is processed into liquefied natural gas. So it includes both terrestrial and marine infrastructures. And the idea here is to try to incorporate biodiversity conservation into development and operation of these infrastructures. So again, we work on the marine side, and basically we're monitoring the key components of the marine ecosystem. So you're going to be hearing from Carlos later about seabirds, which are in high abundance at our site. We look very closely at the fish community as well, the benthos, especially around the hard structures. And then there's also plankton and environmental variables that are monitored. But I want to stress that while this information is really important for the partnership, for our private sector partner, one of the opportunities that's provided to us as researchers is to be able to conduct long-term research on how it seems like some of the species that we see at our site either arrive or are thriving because of the presence of this infrastructure. And so one of those groups is sharks and rays. And you see in this image our one shark species that we have in the record of our survey, and that's the speckled smoothhound. It's actually a critically endangered species. So it's really important for us to understand why these species seem to be congregating around this infrastructure. So if we look at the survey data over roughly the last 10 years, what we see is there's about 5 species of shark. Well, there are 5 species of sharks and rays that have been caught, and it's dominated by one species, the diamond stingray. And if we look at sort of their conservation status, what you see is that all of them are listed as vulnerable, if not the critically endangered smoothhound. And I want to stress that most of these vulnerable, or all of the vulnerable actually categories, just came about in the most recent assessment. Up until then, they were classified as data deficient. So it really highlights how little data we have for a lot of these species. So we set out to estimate residency of sharks and rays surrounding the artificial marine infrastructure, decipher any temporal patterns in detections, and hopefully determine any environmental covariates that might be associated with changes in detection density or frequency. Also to infer movement patterns of sharks and rays around this infrastructure, and hopefully predict occurrence at specific locations using environmental predictor variables. So I'm going to briefly cover the methodology. I've taken out some of like the rudimentary acoustic telemetry slides that I usually present, hoping that people are somewhat familiar with this technique, but feel free to stop me if there's any questions that come up. As many of you may know, there's two main components to an acoustic telemetry system. The first is the acoustic receiver, and we had an array of 3 acoustic receivers. So if you look at the figure on the left, the bullseye shows you where we had our acoustic receivers. Receivers 2 and 3 were moored to buoys, and then the first one was actually attached to a dock. And you can see some of the sampling areas where sharks and rays were caught for our survey within our site. I should mention they were caught using either gill nets or long lines, and any individual that we observed that we thought was healthy enough to tag, we would conduct the surgery to implant the acoustic transmitter. And that basically consisted of making an incision in the ventral side, inserting the transmitter, and then using interrupted sutures to close the incision. So here's sort of a summary of the tagged animals for our study. As you can see, we got a pretty good coverage of the diversity, 4 out of the 5 species that we had in our survey data. And like, the survey data, it was dominated by the diamond stingray. I'll highlight that we tended to focus on some of the larger individuals just to make sure that they could withstand the surgeries. Okay, so I'm going to go over some preliminary results, and I do want to stress the preliminary nature. As I'll mention in a bit, we suffered some equipment loss, and as a result, we only have about 6 months of detection data. So obviously, hopefully enough to get some ideas of what's going on in our system here, but stuff that needs to be followed up with further studies. Okay, so the first thing I want to highlight is the differences in temporal patterns and detections. So just to orient you quickly, on the y-axis here, you'll see a code which has the genus and species, Hippanus dipterourus for the diamond stingray, Mustela mento for the speckled smoothhound. And then obviously the numbers refer to different individuals. So we did have only 3 individuals that we received detections for. However, enough to start to see some patterns. I'll also mention that the open circles represent the tag. So one thing that's really interesting is our speckled smoothhound was, as you see, tagged towards the end of our series of data, but actually had more detections than the other 2 diamond stingray individuals combined. So it was detected extremely frequently for the last 2 months or so of data that we have. And we would say that this species seems to be like a resident at that site, like constantly there. On the other hand, as we look at the diamond stingrays, there's some potential seasonality that we see here. They seem to be detected quite a bit more during summer, austral summer, I should say, in our system. And then if we drill down on those diamond stingrays and look at some of the spatial patterns in detections, quickly I'll orient you. Here we have a contour plot showing relative number of detections. What we see is that we definitely have some difference in the habitat use around this site. It looks like our second individual was spending a little bit more time further from the shore. There's some potential habitat partitioning here. It's something obviously we want to follow up and understand a little better. All right. Excuse me, I forgot to change the translation here, and it says lessons learned. It should say and lessons learned. But anyway, the conclusion is we saw differences in residency and potentially seasonality between species. We also looked at the spatial differences in individuals and saw the potential for habitat partitioning. And as I mentioned, we suffered some equipment loss. This was mainly related to the heavy wave action that we received during the southern hemisphere winter here. So think May through September. And what this means is that we need to have a lot more planning and checking up on equipment moving forward to make sure we don't have that happen again. So our next steps is to deploy additional receivers to both replace the ones that were lost and hopefully expand our array, thanks to some receivers that Matt has been gracious enough to provide for us. We hope to test different mooring designs to really try to key in on what the best mooring would be to make sure we don't suffer any more equipment loss. Increase the sample size and diversity of tagged animals. We hope to get some smaller tags so we can tag some smaller stingrays that occur at our site, and to continue to submit proposals for additional funding. And I was going to take questions, but I understand we're not doing that. So I'll just thank you.
Thank you, Martin.
Okay, thank you very much, Martin. So nice to see you. Our next speaker is Carlos Zabalaga. Carlos, are you there? Are you there, Carlos? Okay, yes, actually, your turn. Okay, okay. Carlos has decades of experience studying the behavior, ecology, and movement of seabirds. He is currently a professor at the Universidad Científica del Sur in Lima, Peru, where he founded the Marine Ecosystem Research Unit, Peruvian Seabird Group, and mentors a laboratory of graduate students dedicated to the study and conservation of seabirds with projects that involve biologging, rodent eradication, fishery interactions, disease detection, and plastic pollution. Welcome, Carlos.
Thank you, Jimena, and everybody for inviting me to this meeting. And I'm going to share my screen. Okay, I am assuming everybody can see it and
Yes.
can hear me. Yes?
Yes.
Okay, good. All right, so I changed the title of my presentation many times. I wasn't very sure what to present. So at the end, I decided to tell a story about the biologging projects I am carrying out in my seabird lab at the university here in Lima, but also how are they connected to we started years ago in Peru LNG port installations that Martin already explained very in detail. And so I will tell you like a general overview, a general story about the research I am conducting and how they are connecting to what we are carrying out in the Peru LNG port. Here. All right. First, it's very important to understand the scenario where the Peruvian seabirds are, living, are breeding, and the Peruvian coast is strongly influenced by the presence of the Humboldt Current. It's this rich cold water upwelling ecosystem, which is recognized as one of the fourth most important upwelling ecosystems in the world. So the main characteristics of this ecosystem is that it's the high productivity first, and all these high productivity converge mainly in one species that is represented here, is the Peruvian anchovy or anchoveta, which is, the key species in the ecosystem that is part of an important food web that end up to top predators as seabirds. And because the presence of the Humboldt Current is, or the Humboldt Current is it's a cold Humboldt. It's a cold current. We got this landscape in the coast. We got a barren landscape with no vegetation, except of course about 36 valleys along the Peruvian coast. But most of the Peruvian coast is a desert, as you can see here in the pictures, where most of the seabirds are breeding on either on the Guano Islands or on protected walled-off headlands. Here you can see, for example, some pictures of Guano Islands that are the breeding or the most important breeding sites of many species of seabirds in Peru. Actually, we got 12 endemic species of the Humboldt Current, I mean seabird species. We also have 6 extra species that also breed in the Peruvian coast, but they are not necessarily endemic, but they also breed in other sites. And also we have 71 migrants. They are coming from different parts of the world, mainly from North America, from New Zealand, Australia, Antarctica, South Africa, and part of them from the Caribbean too. So I'm talking about 71 seabird species. And this is something important to mention because under the scenario of the avian flu, knowing the pathways of the migrants from North America is very important to understand the origin of the mass mortality of different species of seabirds that occurred late last year and early this year. So I think the connection and understanding of the movement of migrants from North America to South America and vice versa will help us to understand better, the spread and of this disease. As an introduction, I would like to present you some of the most representative endemic species of the Humboldt Current. So we got the Humboldt penguin, the Peruvian diving petrel, the Peruvian tern, Peruvian booby, the Inca tern, the Belcher's gull, the one-eyed cormorant, the Peruvian pelican, the wedge-ramped stone petrel, the hornbill stone petrel, and the Markham's stone petrel. These are the most representative endemic species that, of course, breed on different sites along the Peruvian coast. All right, so what have we done in the last 7 years in our lab at the university in Lima is basically a lot of biologging work for different reasons. We wanted to understand if seabirds can be used of bioindicators of ocean health in terms of, for example, anthropogenic pressure, also climate change, El Nino events, and recently the movement patterns during the avian influenza outbreak. Here are some examples of how we use different instruments, mainly data loggers that carry GPS, time depth recorders, accelerometers, and temperature sensors. So this is an example of the movements of one-eyed cormorants, and one of the sites we are monitoring since 2017. It's the one-eyed cormorant and how they use the marine habitat for feeding and what are the home range of these animals to during the breeding season. As you can see here, is very variable among years, so they can change direction and also the area where they are feeding. And our main question was why these animals are modifying their foraging strategies. And we ended up with different answers. I am showing you part of it on how the cormorants are basically moving according to sea surface temperature, and which is of course related to the presence of anchovies that prefer cold waters instead of warm waters. And so we relay this information not only to oceanographic variables, but also to breeding responses that birds are having on land, either with breeding success, growth of chicks, and other variables that are important to understand how good they are doing on different environmental scenarios. Here, there are overlapping between the foraging areas and sea surface temperatures obtained for satellite imagery, and also how what is the distribution of the sea surface temperature, of the temperature of the sea in the water column. This is also important because it determines how deep or how shallow these cormorants are diving, and this was possible because the data loggers are carrying temperature sensors with quick response, and while the cormorant is diving, so it can record every second the temperature of the environment. So this is also helping us to understand better why these cormorants are feeding in some species of fish, but also to understand why they are spending more time and spending more energy when they are looking for their food. Another example of biologging work we are doing is to attach small video cameras together with GPS and depth recorders to Humboldt penguins, also at Punta San Juan, and this is southern Peru, to understand some strategies when they are capturing their prey. Here in some images, you can easily see how these birds are, going to the bottom of the school, fish school, and then depolarizing the school, breaking it apart, and then, grabbing the fish from below, which is the most common strategy they use for capturing their prey. After joining the data from the video cameras, the time depth recorders, and the GPS, so we can have a better idea of what is going on underwater. Here is a small representation of what I'm saying. This is the diving depth in the y-axis, and here is the time, local time, in the x-axis. And this is the diving profiles of the penguins. And here in each dot is represented by the prey capture. How do we know this? Because the video cameras are synchronized with the data loggers. And so we can easily see and check what are the strategies they use to herd the schools to the surface. And in this way, they can capture the prey more easily.
Carlos, just noting that we're over 10 minutes. You can.
Excuse me? Sorry.
We're over 10 minutes now.
Oh, okay, sorry.
That's okay.
And this is another example of the fishery, of the biologging work we are doing, and to understand the interactions between seabirds, in this case, the Peruvian boobies, and the fisheries for commercial fisheries for anchoveta. And here you can see in the images how the birds hunt or fish underwater inside the pure seine, and also how they interact with other species of seabirds when the fishermen set the nets. In some instances, we can also check the name and the number of the fishing vessels, so we can double check if they are not fishing in illegal zones or no-take zones for fishing, for example. And we can also overlap this information to other free online platforms like Global Fishing Watch to understand better the kind of interaction and how this interaction with the fishing vessels are related to the fishing effort that the vessel have. Finally, how we bring all this experience into the BMAP project we have in the Peru LNG installations, as already Martin explained, is to use video cameras and data loggers in humboldt penguins that are using the breakwater. Here, as you can see in the photographs, the penguins are nesting, are using the installations, the breakwater for nesting, because there are a lot of crevice that the humboldt penguins use for nesting. And the idea, we haven't done this yet because the penguins are not in large numbers now because El Niño event. And but the idea is to compare how they are performing, in terms of foraging behavior in an area that is protected and is excluded by human disturbance in comparison to a natural breeding area. And for this, we are going to collaborate with another program here in Peru to do a simultaneous tracking of the foraging behavior of humboldt penguins. We also started a tracking work of cormorants. This is neotropic cormorants in Peru LNG plant to use the cormorants as oceanographers. And for doing this, we use a different technology. They are data loggers using GSM technology to provide information about the salinity, temperature, and temperature profile in the water column. And this is done in collaboration with the Cormorant Oceanographic Project from Oregon State University. And here, I'm just showing you an example on how the cormorants are doing now, the present day. And they are basically inshore feeders, but they are collecting a lot of information, not only about their movements, but also about the ocean they are using for feeding. Thank you very much.
Thank you. Squeeze in. Do you want to kick us off, Matt?
Sure. Yeah, and I just wanted to thank all the speakers again. Really interesting work, and we're a little bit concerned about time for discussion because we know it's so much exciting things going on. But thank you again to all the speakers, and I guess I want us all to be thinking about Smithsonian is in the process of a couple of years of where we're launching Life on a Sustainable Planet initiative that is sort of a framing device for a lot of the science and conservation work that we're doing. And oceans are a big part of that. So that was part of the motivation for bringing the speakers together today. And thinking about ways that we can work together more as that program develops more and look for opportunities to work together. So I guess my question that I've been waiting to ask Alex is, where Martin presented some of the work and Carlos going on in Peru, and we're interested in sort of thoughts on what you see as sort of opportunities potentially for us, for Autumn, for others to potentially build some more collaborations with Miramar. So there's Hector's work, but I think there's a lot more opportunity as well.
Absolutely. I've been sitting listening to a lot of the presentations thinking the same. I mean, at the moment in this area, we're looking at developing a kind of a potential roadmap for the next phase of marine conservation. And so we're putting together all the information that we have on the movement ecology of a bunch of different species across all taxa. Seabirds is something that we don't have a lot of information on. There's work done on albatrosses and the Galapagos petrel, a little bit of frigate bird work, but I'd not seen a lot of the information that was just shown now. So I think pooling data sets and doing cross taxa studies of where hotspots or movement areas might be would be a first. The other thing is more hands-on field work, expeditions. Any work at sea is costly to get boat time, and so sharing boat time is always very useful. And that's just scratching the surface. So I'll throw that back at you and open it up.
Yeah, I mean, I think, we're sort of, I've mostly been working with Martin and Jimena at that side on the species Martin was talking about. But I think there's a lot of interest in other places. I don't think we have any representatives here today, but SERC's Marine Invasive Species Program is doing a lot of work in Galapagos too, which you may be aware of. So that there's certainly a lot of connections with that area, and so I think a lot of opportunity to think about what proposals we might write together, or what questions we might want to address together. And I can let Autumn or others speak to the syntheses of seabird data and other things that are passing through that region too. But I think I'm hoping to pass off to Jimenez some more receivers and tags today. Or they will soon be going to Peru to expand that project.
Yeah, I totally agree that there's and I'm just gonna quickly say this before passing to Martin, but I think there is a great opportunity for collaboration and maybe expanding the research to other taxonomic groups. I know, Alex, you've been working with large animals, sharks and whales and sea turtles. But as you've seen Martin's work and with rays, which is like a really underrepresented group, but really important as well. And I would think that maybe that's an area of opportunity to expand the research. And they're definitely less costly than going sharking and tagging sharks out there in the open ocean, I would say. But definitely an area for expansion and more research, as well as the seabirds. We're just scratching the surface, and Carlos is leading the research in Peru, and great opportunity to understand more about animal migration in general. And we're pretty much concerned with the avian influenza, not only on seabirds but now in mammals as well. And so yeah.
Yeah, and one of the things that's kind of been at the background of a lot of conversations that we've had, people like David Anderson over at Wake, who study albatrosses, and the shark people and sea lion people, is these lower trophic level species and what's driving their movements, because we're assuming a lot of the movements of the top predators are related to foraging, and that's something that we've not been able to really address. And looking at certain other areas of the world that have kind of big acoustic survey monitoring programs, but finding some way, I think, to understand those movements of these, the anchovetas and the Pacific sardines and the flying fish. I don't know how you do that, but it's something I think would unite a lot of the different behavioral ecologists across taxa.
Yeah, that's something that people here are thinking about a lot as well. That's part of the motivation of the alewife tracking, but those are still 30 centimeter long fish. But there's a lot of interest in some of the smaller menhaden and anchovies that we have here too, that are critical forage for a lot of things. And it's potentially an area to think about a lot as a potential collaboration, I think. And I did want to mention that we did invite Hector to the meeting today too, but just so everyone's aware, he's out on a boat tagging blue sharks today. So, well, hopefully tagging blue sharks. So he wasn't able to join for this discussion. Martine, you have a question?
Yeah, I guess I just wanted to mention as something that occurred to me as something concrete we could try to start doing. And I know Alex mentioned that they did a lot more acoustic work in the early days. I'm not sure how much is going on now, but from what we could tell, interacting with Ocean Tracking Network, the only node for us to incorporate in our region is Migramar. So we've actually, our species that we're tagging currently don't seem to be migrating great distances, at least from what we know. But it'd be good to kind of start that process of incorporating into the same node for sharing data for when we are able to tag more highly migratory species and be able to share detections across borders. So anyway, that we've already started to put together our metadata in the OTN network format. And I guess I was just curious what Alex thought about us maybe sharing data through that node.
That's what it's for. The Migramar OTN node, it was for the region. You don't have to be a member of Migramar. It was just a service. It was, in essence, it's trying to promote more of these studies, because the wider coverage of receivers we have, the better information we're going to get. So yes, absolutely. We can maybe touch base afterwards to figure out how to get the data in. But yeah.
Awesome. Thank you.
Carlos, did you have a comment?
Yeah, sure. I think the connectivity of both hemispheres in terms of, seabird pathways is pretty obvious. And Otto Lin showed very clearly, the movements of three species of jaggers and Arctic terns. But in North America, there are other species that can also be tagged for understanding better understanding this connection. We have, for example, elegant terns, royal terns, sandwich terns, laughing gulls, Franklin gulls, etc., that can also be studied, tracked from North America, and see how the pathways of these seabirds are connected to South America. I think that we can widen it could be, collaborative work between Peru in particular and North America. I think this area in terms of animal movement will be very interesting, particularly under the avian flu scenario.
And thank you, Carlos. I agree, and some of those species have been tracked and some of them haven't. And BirdLife International Seabird Tracking Database is a good place to start to look for some of those tracks. But getting organized with why we're requesting them from other researchers and bringing them together, I think would be really useful for this region. I haven't really seen that done as much in that region with the migrants from North America. I see a lot of syntheses from the Southern Hemisphere and the endemic nesters, but not as many that are coming from the North. So I think that would be really valuable and something that we could work on together for sure. Fun to see. And some of the Gobi workshops I've attended, I saw Gobi's logo on Alex's slide. We're kind of getting at that with these regional workshops that we're bringing together data sets, but they tended to be who was in the room that had data sets, and BirdLife was at some of them and not at others. And so I think a more concerted effort is definitely necessary, and to see what species still within the Humboldt Current could use some additional data, and some field studies would be great too. Yeah, yeah,
Yeah, so I think we should probably wrap it up here, but I really appreciate this, abbreviated discussion. I think there's a lot that we can all follow up on. And so I'm again, thankful to everyone for joining today, both online and in person. And wanted to highlight, I guess, one other announcement I forgot to say at the beginning is I'm going to be giving CERC's public evening lecture on Tuesday next week, Tuesday evening, I think it's 7 p.m. About some of our work on migrations of cow nose rays from Chesapeake Bay to points beyond. I don't want to spoil the story. So I'll be doing that as well on Tuesday and make sure that Lacey has the information and can forward it around to people. But unless there are any other announcements, I think we're ready to say goodbye to folks.
Yeah, and just wanted to thank Matt and Kim for hosting and making everything run smoothly. So, and our visitors here, Amanda and Ronaldo, and also online. Thank you everyone for joining. One of my favorite things about Movement of Life is sort of our global reach and ability to connect. So thank you. All right.
Great, thank you, everyone.
Thank
And
you.
for the in the room, I figure we can do a quick round of introductions. You know who's out here. We did